Good afternoon, everyone. Imagine your immune system as a library. Most books are printed once and never change, but one special section rewrites itself every time you get an infection. That section is your antibody genes, and the librarian doing the rewriting is Tasuku Honjo. Born in Kyoto in 1942, Honjo earned his MD and PhD from Kyoto University. We all know he won the 2018 Nobel Prize for discovering PT1. But today, I want to take you back 2 decades earlier, to 2002, when he asked a question that was just as revolutionary. How does the immune system switch antibody classes without changing the story it's telling? As you'll see, he didn't just discover a molecule, he uncovered an entire editing system that lets our genome rewrite itself on demand. Let's set the scene. When the B cell first meets its matching pathogen, it produces IgM antibodies, big, pentameric, good at agglutinating bugs, but not great at fine-tuned jobs like crossing the placenta or triggering mast cells. To do those jobs, the B cells has to swap the constant region of its antibody gene, switching from IgM to IgG, IgA, or IgE, while keeping the antigen binding site exactly the same. That swap is called class switch recombination, or CSR. The bold question Honjo posed was, CSR looks like a DNA recombination event, and somatic hypermutation, SHM, looks like a DNA repair event. Yet both occur in the same B cell at the same time. Could one molecular machine control both? Answering that means diving into the DNA level, looking for a common editor that could cut, mutate, and paste antibody genes with surgical precision. Here's where the story gets electrifying. Honjo's group had already cloned a mysterious enzyme, impressed only in germinal center B cells. They named it AIDE, activation-induced cytidine desaminase. In the 2002 Annual Review of Immunology paper, Honjo, Kinoshita, and Muromatsu sewed together every loose thread and proposed the following three-step model illustrated in Figure 1. To test this model, Honjo's team used artificial Swiss substrate in culture B cells and 8 knockout mice. The result was categorical: no AIDE, no CSR, no SHM, proving AIDE is not a mere accessory. It is the chief editor. So what did Honjo really uncover? First, AIDE is essential. It sits at the crossroad of antibody diversification. Second, DNA secondary structure matters. Palindromes aren't just repetitive drug. They are molecular zip codes guiding AIDE-dependent cleavage. Third, transcription level equals mutation rate. The more actively a gene is read, the more likely it is to be rewritten. A principle now recognized genome-wide, yet the story is far from finished. We still don't know the exact RNA or DNA that AIDE deaminate first. Are there other AIDE targets outside immunoglobulin loci? Could misregulate AIDE explain B-cell lymphomas where off-target mutations accumulate? And if we could dial AIDE activity up or down pharmacologically, might we enhance vaccine responses or dampen autoimmune antibody production? These questions are driving labs around the world today, all building on the mechanistic blueprint Honjo dropped 2 decades ago. To conclude, Tasuku Honjo didn't just explain how we make better antibodies. He revealed that our genome is a living document, constantly revised by an RNA guide editor named AIDE. His work reminds us that revolutionary science often begins with a simple, audacious question and a willingness to rewrite the textbooks, one nucleotide at a time.